We're in the middle of this absolutely massive biological revolution, but it's not happening in some giant flashy lab. It's happening inside every single cell in your body. Today, we're diving into the data explosion that is fundamentally changing how we understand life itself.
So think about this for a second. What if you wanted to understand a really complex disease like cancer? For decades, the only way was to, grind up a piece of tissue and look at the average genetic signal from all those cells mixed together. But what if you could zoom in, way in, and profile every single cell, one by one? What secrets would you find? Well, that's not science fiction anymore. That is the incredible promise of single-cell analysis. To really get a feel for this, let's use an analogy. Think about a fruit smoothie. The old way, bulk analysis, is like tasting the finished smoothie. You get the average flavor, right? It's pretty good, maybe strawberry banana. But the new way, single-cell analysis, is like getting to taste every single piece of fruit before it goes into the blender. You'd know, wow, this strawberry is super ripe, that blueberry is a little tart, and this piece of banana is just perfect. You understand each unique part, not just the blended mush. That's the gigantic leap we're talking about. So here's where we're headed today. First, we'll look at this new kind of microscope. Then we'll face the absolute tsunami of data it creates. We'll meet the amazing tool that organizes all that chaos, walk through the process from raw numbers to actual insights, and then we'll see the incredible biological secrets this is all unlocking. And it's all powered by an open source future. Let's get into it. All right, first up, let's just take a moment to appreciate this new tool. It's kind of like a new microscope, but instead of using lenses, it uses genetic sequencing. This is the technology that lets scientists finally get past that blurry, averaged-out picture and see the crisp, unique identity of every single cell. It's revealing a level of diversity inside us that we just never knew existed. But here's the catch. When you can suddenly see everything all at once, you create a whole new kind of problem. This incredible power doesn't just give us answers. It unleashes an absolute tidal wave of data. And the scale? It is just mind-boggling. In just one experiment, a scientist can now measure millions of individual cells. Every single one is its own unique data point. And if that wasn't enough, it gets even more complex. For each and every one of those millions of cells, we can measure thousands of different things, like the activity of all its different genes. So suddenly, you're not just looking at a big spreadsheet. You're looking at a data set with millions of rows and thousands of columns. It's immense. This data tsunami really boils down to two huge challenges. First, there's the sheer scale of it all. It's just an enormous number of observations to process. And second, there's a tricky problem called sparsity. A lot of the data has gaps or zeros where there should be a measurement. It's kind of like having a puzzle with tons of missing pieces, which makes the analysis really, really difficult. So, how on earth do you even start to make sense of this? You need a system. You need a fantastic organizer. And for thousands of scientists all over the world, that solution has a name: Bioconductor. It's the tool that brings some much-needed order to all this cellular chaos. So what is Bioconductor? Well, at its core, it's a huge open-source software project. It's basically a massive collection of tools, all written in the R programming language. And they're all specifically designed to handle the gigantic, messy data sets that come out of modern biology. And this quote right here really gets to the heart of why it's so successful. See, it's not just a bunch of random tools thrown together. Its biggest advantage is that it creates a common language for the data. It provides a standard way to represent and structure all this complex information so that different tools made by different people can all talk to each other and work together perfectly. And the heart of that whole system is a special object called the single-cell experiment. The best way to think about it is like the ultimate digital filing cabinet. It's brilliant. It doesn't just hold the raw data, the gene counts. It has separate drawers for everything else: information about each cell, information about each gene, and even a place to store the simplified patterns you find along the way. Everything stays neat, organized, and connected. Okay, so that all sounds great in theory, but how does it actually work? How does Bioconductor help a scientist go from that chaotic flood of numbers to a clear, meaningful biological discovery? Well, the journey pretty much follows these 5 key steps. You start by just getting the raw data in. Then comes a super important step: quality control, cleaning out all the junk. Step 3, normalization, is all about making sure you can compare apples to apples across different cells. Then the really cool part, using powerful algorithms to find the important patterns hidden in all that noise. And finally, you can integrate data from lots of different experiments together to see the really big picture. So after all that work, the organizing, the cleaning, the processing, what's the big payoff? What do we actually get to learn? Well, this is where it gets really exciting. This is where we unlock some amazing biological secrets. One of the first things you can do is called clustering. Basically, smart algorithms go through the data and automatically group cells together based on what their genes are doing. And just like that, a hidden structure starts to appear. Suddenly, you can see all these distinct populations of cells you couldn't see before. These are immune cells, those are skin cells. And hey, what's that tiny group over there? It could be a rare type of cancer cell no one's ever been able to isolate. But here's a really, really important point to remember. A cluster that the computer finds is just a statistical grouping. A cell type is a real biological thing. And as this quote says, they aren't the same. Clustering isn't the final answer. It's a tool, just like a microscope, that helps you explore the data and get closer to the biological truth. And it gets even cooler. We can go beyond just taking a snapshot of what cells are. We can actually map out how they change over time. It's called trajectory analysis. The computer orders all the cells along a continuous path, which basically lets us watch a biological journey unfold, like watching a single stem cell slowly decide it's going to become a brain cell. You can really picture it like a developmental roadmap. Instead of just seeing the starting point, the stem cell, and the destination, the specialized cell, trajectory analysis lets us see all the stops and turns along the way. We get to watch the entire fascinating journey. And this brings us to what might be the biggest idea of all. None of this incredible science would be happening this fast if it weren't for a powerful philosophy at its core. The future of science is open, and it's built on collaboration. And you can literally see this collaboration in action. Just look at the growth. The number of special software packages available in Bioconductor has just exploded, going from less than 50 back in 2010 to well over 300 only 8 years later. This isn't one company building a product. This is a global community of scientists building tools for each other. The power of this community approach is just huge. It makes cutting-edge science accessible to any researcher anywhere in the world. It helps ensure that scientific results are reproducible, which is the absolute foundation of good science. Because of that common structure we talked about, different tools just work together. And most importantly, it creates this global network where people are constantly sharing code, helping each other out, and building on what's come before. So let's bring it all home. Bioconductor is like the conductor of a massive orchestra. It's orchestrating the analysis of millions and millions of cells. And in doing so, it turns that overwhelming tsunami of data into clear, verifiable, and often totally groundbreaking biological breakthroughs. We are now at a moment in history where massive projects are underway to literally map the entire human body one cell at a time. The tools are here, the community is building them faster than ever, and the data is pouring in. So the only question left is, what amazing mysteries are we going to solve next?